Hi everyone, hope you all doing well. Stay home, stay safe and learn chemistry on your fingertips. Here in this video, we are going to learn the very important qualitative tests of carbohydrates, and we know that carbohydrates are one of the major energy sources for living beings on earth. That's why we can see frequent questions from carbohydrate tests in various chemistry examinations, and hence this video is worthwhile to watch completely. And one more thing, I'm very happy to inform you that we have taken a new version of mobile app, that is chemmasters.online, which has all the user-friendly features and where one can get all the chemistry courses with the best content on demand. And here we are offering an opportunity for chemistry educators on chemmasters.online. If you have student reach around you, and you can contact us for more details. Okay, right. So here you can download chemmasters.online from Play Stores of your mobile. So you can watch free video lectures of various chemistry courses, and if you are interested, you can join the courses to access live and recorded lectures along with mock tests, practice sessions and doubt sessions, study material and many more to get guaranteed success. Thank you very much. Now we are going to enter into the lecture. So here the learning goals of this video. So we are going to learn qualitative tests of carbohydrates briefly, and you are going to get table form of all the tests, and you can get quiz on these carbohydrate tests. Okay, right. The first test here we are going to look at Molisch test. So Molisch test is a specific for all the carbohydrates. That means we can identify the carbohydrate from a given sample. So if the given sample has a carbohydrate, then it gives positive test with the Molisch test or Molisch reagent. So by just identifying a purple ring. So just look at the test tube. Okay. Here monosaccharides give a rapid positive test, while disaccharides and polysaccharides react slower. Do you know the reason? The simple reason is there, that is disaccharides and polysaccharides get hydrolyzed first, then they can react with Molisch reagent. Okay, so that's why here disaccharides and polysaccharides react slower. So whenever pentoses react with concentrated H2SO4, we know that the concentrated H2SO4 is a dehydrating agent, and it dehydrates pentoses and produces furfural. And that furfural reacts with alpha naphthol, the Molisch reagent, and if it gives purple color, so simply it is a carbohydrate. Okay, right. And whenever hexose is dehydrated with H2SO4, they are going to produce 5-hydroxy methyl furfural. Then 5-hydroxy methyl furfural reacts with alpha naphthol, and it is going to produce purple colored ring. Okay, so what is the difference between pentoses and hexose here? Pentoses are going to produce furfural, a simple furfural, whereas hexoses are going to produce 5-hydroxy methyl furfural. Okay, now the next test is Anthron test. So this test is specific for all the carbohydrates, so as just like Molisch test. Okay, so monosaccharides gives a rapid positive test, disaccharides and polysaccharides react slower. So the same reason we can give or we can apply here as we given for that Molisch test, because disaccharides and polysaccharides must get hydrolyzed before going to react with Anthron reagent. Okay, so here the carbohydrates, after dehydrating with H2SO4, they are going to produce furfural derivative. Then furfural derivative reacts with Anthron and which is going to give blue-green complex as shown in the test tube. Okay, so what is the major difference between Molisch test and Anthron test? Both are going to identify carbohydrates. Okay, if the sample is carbohydrate, so that is going to give positive test for Molisch and Anthron reagents. So the major difference is Molisch test uses alpha-naphthol, Anthron test uses Anthron. Okay, now the next one is Benedict test. So Benedict test detects the presence of reducing sugars of the carbohydrates. Okay, so all monosaccharides are considered as reducing sugars because monosaccharides having a free reactive carbonyl group. So that carbonyl group may be aldehyde or keto functional group. Okay, so if a carbohydrate has a free reactive carbonyl group, then it is going to give positive test for Benedict's reagent. Okay, so simply Benedict's test is used to identify the presence of reducing sugars. Some disaccharides have exposed carbonyl groups and are also reducing sugars. Can you give the example for the disaccharide which give positive test of Benedict's? Okay, so we can see in the next slide. Except sucrose, so sucrose is considered as popularly considered as a non-reducing sugar. That is just because of the absence of a free carbonyl group. So there is no free aldehyde or ketone functional group in sucrose. That's why sucrose is going to give negative test for the Benedict's reagent, and it is considered as non-reducing disaccharide. Okay, so large polymers of glucose, so here we can give the example starch, are not reducing sugars. That is just due to the concentration of hemiester group is very low. So that's why they are not going to give a positive Benedict's test. Okay, reducing sugars are simply oxidized by the copper reagent of this Benedict test in solution to form a carboxylic acid, and it is going to give reddish precipitate of copper oxide. Here copper is reduced. Okay, copper plus 2 is reduced to copper plus 1 oxide. Okay, right. So here the lab tests are taken. So if you take glucose, sucrose and lactose, we know that glucose is a monosaccharide, and it is having free aldehyde functional group. That's why it gives positive test, and which is going to show a reddish precipitate. Whereas lactose is a disaccharide and it is also given positive test of Benedict's reagent because it is going to produce reddish precipitate because it is having free free functional group. Okay, free carbonyl group. And here the sucrose test is there. So Benedict's reagent is initially blue in color, and so if you take the sucrose in it, there is no change in the color. That's simply indicating that sucrose is not reacting with Benedict's reagent, and that's why it is a non-reducing sugar because of the absence of free carbonyl functional groups in sucrose disaccharide. Okay, now Barfoot's test. So which distinguish between mono, di and polysaccharides? So simply Barfoot test is used to identify monosaccharides. So Barfoot test used copper plus 2 ions in slightly acidic medium, whereas in Benedict test we can use copper plus 2 ions in alkaline medium, whereas in Barfoot test we can use acidic medium. Okay, so reducing sugar whenever reacting with copper plus 2 in aqueous solution, and it is going to produce reddish precipitate. So whenever the sugar is going to give reddish precipitate with Barfoot's test, then we can say that sugar is a reducing sugar. So based on the rates of reactions for mono, di and polysaccharides, so simply here we can say we can identify the we can differentiate mono, di and polysaccharides with the Barfoot test. Okay, so you can see the color in the test tube, reddish precipitate. Now next one is Biol's test, which distinguish between pentose monosaccharides and hexose monosaccharides, which is very important. Okay, so Biol's test uses concentrated HCl as a dehydrating acid and arsenol plus a trace of ferric chloride as condensation reagent. So whenever pentosis reacts with Biol's reagent, they produce bluish or green product, whereas hexosis produces muddy brown to gray product. So based on observance of color, we can identify the given sugar is a pentose or hexose with the Buell's test. Okay, so you can see the test. You can see the test tubes with the colors here. Okay, now the next one is a Sullivan's test, which distinguishes between aldoses like glucose and ketoses like fructose. Okay, fructose here. Now, Sullivan's test uses concentrated HCl as dehydrating agent and resorcinol as condensation reagent. So you can see the difference between Biol's reagent and Sullivan's reagent. In Biol's reagent, we have used arsenol plus traces of FeCl3. Okay, here we are using a resorcinol as a condensation reagent. Okay, now whenever ketohexosis reacts with Sullivan's reagent, then it is going to produce a cherry reddish product, and aldohexosis whenever reacts with Sullivan's reagent produces yellow to faint product. So based on the color differentiations, we can identify ketohexosis and aldohexosis with Sullivan's test. Okay, so here is the table, and it is just look at all the tests are there with the reagent and color changes and identification of carbohydrates. So in Molisch test, we are using H2SO4 and alpha naphthol as reagent combination, and so if you see purple ring on the test tube, then it simply indicates that the presence of carbohydrate. And in anthrone test, we are using H2SO4 and anthrone combination as a reagent, and the color change is blue green, and it is indicating that the carbohydrate is present in the given sample. Now Benedict test, here the reagent is aqueous copper sulfate in alkaline medium, and the color change is reddish precipitate. If you observe reddish precipitate, then the carbohydrate is a reducing sugar. Now Barfoot test, here the reagent is aqueous copper plus 2 in acidic medium, and it is also going to give reddish precipitate, and we can simply say that that is a monosaccharide. And we can also differentiate mono, di and polysaccharides based on the rates of the reactions. Okay, now Biol's test, the reagent is concentrated Cl arsenol and FeCl3. Here arsenol and FeCl3 used as condensation reagents, and the color change is bluish green to muddy brown to gray. So if you observe bluish green, then they are pentoses. If you observe muddy brown to gray, then they are hexoses. Now Sullivan's reagent, Sullivan's test, which uses concentrated HCl and resorcinol as reagent, and color change is cherry red product, and which distinguishes aldoses and ketoses. Okay, so thank you very much, and if you are interested, you can download the mobile app chemmasters.online from Play Stores, and don't waste the time. So you can get the best features from that app, and from where you can get the best chemistry courses, various chemistry courses. Thank you very much. Contact us for any further details. So please don't forget to like, subscribe and share this video, and more, and we can also give the best content here also. Okay, thank you very much.